diagnosis. Now I will give the floor to Professor Salvatore. He's going to speak about the most important general aspects of predictive genetics.
English to thank very much the English speaking people here, very outstanding scientists which have honored this session. But since my broken English is worse than that my broken Italian, I would speak in Italian with English slides. Grazie, Dottor.
I would like to thank Dr. Giuliani and Senator Danna for inviting me. I am very grateful for being here. I changed the title of my presentation because there will be illustrious scientists who will speak about genetics, and also they will speak about the human genome as predictive medicine. That was treated by Dr. Guerrieri. So we'll speak about some other aspects, and also I will show you the results of some tests that we carried out in my laboratory in particular. So, we are now metabolic phenotypes, and we are also subject to a series of variations that can be physiological, as we are used to saying, or they can be pathologies. When there's a disease, when there's the onset of a disease, you can see many arrows that are green and red that go towards this metabolic phenotype, and it's two possible and current variations. Some of them are genetic, which means that they come from the pool of genes. I will choose your gene pool, one could say, from your parents, but clearly that's a joke. This is not possible, and I was just joking. But as the previous speaker said very well, reading the human genome, that is the sequence of genes, is smart. Is a smart thing to do. We're not only interested in the gene setup based on what we know in those genes, and we know little for now about them, even if science is making huge leaps forward in the last years. But the human genome recently, in the last 20 years, so not that recently, let's say, that has acquired another series of genomes that accompany it, starting from the birth, but even before birth, as we've seen. This is called, generally, the human microbiome. That is not human, starting from the bacteria that are part of our body, but they accompany us since the womb. So clearly we have to take that into account because they contribute and cooperate closely with the functions of our genomes. And so I will speak a bit about those aspects also because, as I will say, we have worked a bit on that and we've had some results that seemed useful from this perspective. Epigenetics, of course, with the various definitions of this branch of science that is very important, they contribute very much to the DNA analysis, because DNA is like a foolish entity that is there and does not know what to do, and it needs other factors that determine its behavior and development. So epigenetics has a great influence, as it was said. Here as well, there are red and green arrows because they can be positive and negative for the physiological homeostasis of the body. And then there's a series, we don't get into detail here, but I will touch upon those aspects too. I asked the president to warn me when my time is about to run out, because very often I speak for too long. And I should stop when it's time to stop. But here you can see that there's a great factor that are the drugs, the medicines that have a good effect, a positive effect, but they could also have a negative effect because they all have side effects that vary in the different persons, and genetics can help greatly to understand through pharmacogenomics the positive or highly negative effects. Some therapies are even forbidden without a genetics test that has to be carried out in advance, because some effects are even deadly in the case of some virus diseases. So moving forward, let us now speak about the microbiota together with the human genome makes of each one of us a superorganism, because as we are closely linked with bacteria that operate in our intestine. In the last years, this was called a forgotten organ because it was, let's say, neglected in the past. And this organ is accompanied by a series of bacteria that are revealing very relevant secrets that help us understand some human diseases. We could even say that the excess of interest for this field has led to this. Almost all diseases, neoplasias and virus disease and even others, are influenced by a presence or an absence of a series of microbes that carry out some functions. They are very useful for our organism, for our body, because they produce very useful substances such as vitamins, amino acids, essential amino acids. They can detox negative products that we get when we eat, and they become part of our body, and so they create the best physiological conditions. And so there's a mutual benefit here, because also they are very healthy when we are both healthy, when we are healthy too. So here you can see that, as I said very briefly, we used to think that we were born totally sterile, and that could be true, but we are immediately colonized from our births, but also in the womb. There are the first signs of presence of bacteria that come from the mother, of course. This intestinal microbiota, that is the main component, it's just a small percentage of the other microbiotas that are open to the outside and that are on the surface of our organism. That is the main one. And the number of cells of bacteria, of course, bacterial cells that are present in the intestine are even like the ones that had been counted that are 10 to 13. So these are even equal or even higher. We have taxa from a phyla to species. They are tens of thousands, and of course they contain a certain amount of genes that are more numerous than the genes that can be templates of proteins, and even 3.3 million of genes. So, as for, I will jump forward because time is running out, but even from the womb of the mother, there's a bacterial contamination that reaches even the fetus tissues, and so this leads to some cell and molecular mechanisms that are physiological or pathological mechanisms that then continue or develop over time during our life up until the time when we are old. One of the most important systems of our organism, that is the immune neonatal system, is very much influenced by the presence of those bacteria that exist before birth, and so they're very important. Much more recently, this term was created, holobiont. It was created by a group of Israel, and it shows very well that our body has to be considered more and more as the combination of the microbiota with our genome. And the genome should be considered as the starting point for all mutations that happen later on. And from the anatomical, metabolical, and immunological point of view, it acts especially during the development and the evolution. Our human genome is highly conserved, which means that the speed of its mutation cannot be compared to the mutations that happen in the microbiome, where the speed of changes is very high, and it reacts to the external environment. Also, the horizontal gene transfer mutation is very high compared to what happens in the human genome. There's also the microbial core. According to some scientists, the core is equal or similar to an organism, and it can be conserved in the cases of host generations, and it was measured in the human residues of many centuries ago. This part is called microbial core, that survives to the changes that happen in the environment, in the surrounding environment, that have happened over time, over centuries. As Darwin said, it is not the strongest species that survives, nor the most intelligent, but the one that is the most capable to adapt to changes of the surrounding environment. Just to give you an example of a research that we carried out some years ago in our laboratory, here we can see a case report, to be more precise, concerning Crohn's disease. The yellow column shows the situation of the bacterial population. Here's the Shan index. I will not get into detail here because time is running out, but the yellow column is the situation of the patient. That was a patient who was 13 years old, while the green and blue columns show the concentration of a phylum, and the other taxa, microbic taxa, they are equal to those after the therapy. So this is a good example not only of the physiopathological variety that we can have in a pathology, but also shows that it is possible to revert this trend and carry out a very significant therapy regarding those bacteria that can then solve this microbial diversity that in this case had an impact early also on the health of the kid, of the child, sorry. This is something concerning the celiac disease that is becoming more and more common. 1 or 2% of the population suffers from it. And here again we've found some novelties, that is the presence of a flavescence and of a Neisseria flavescens. It was not found in the. It was found in the duodenal mucosa, and some pediatrists cooperated in this study, and here you can see that starting from phylum up until the gender and the species, we can identify the presence of bacteria This led to the discovery of viral triggers with beautiful studies carried out by other American laboratories. And so it was found that there's viral or bacterial triggers, and also some bacteria were studied at the same time. So we could contribute to the etiopathogenesis of the disease. It is a disease related to the presence of gluten in the diet, and we can use some specific markers, HL2, 8, and 7. So moving forward, here we can see a hyperinflammation in the man. And here we can see also the migration in the isosoma, and that is different when there's the Neisseria. Here we can see that this was shown by another study, the relationship with 18S and 16S, one is for fungus and the other is for bacteria. But in the case of fungus, there's no difference. So it shows that there are some specific bacteria that can be triggers for this disease, and they can, and this result can lead to some therapies, we hope. The most interesting thing is something that has not been published yet, and I'm sorry for that. It shows clearly that in the oral pharynx we can find the same area that we found in the duodenal, only amongst a few patients. That is clearly sampling should be more extended. And we can see that that starts from the oropharynx. That these are the indexes. I just wanted to say something, because it's so that in the audience there are very good biologists that deal with the forensic aspects. The microbiome can be a forensic toolkit. We know that forensic biology contributes to many aspects of our social and also non-social life. That is a joke, sorry. But the use of the microbiome is a novelty, and this can lead to a geolocalization. So, of course, we shouldn't take the sample of a cigarette to see the DNA, but we can study the microbiome to understand where a person has been. So the set of those qualitative and quantitative. So we can have a qualitative and quantitative analysis here through higher sequencing based on ribosome. We can study these aspects. Clearly, this is the technological base to identify those bacteria, and through those bacteria we can have a geolocalization of the person and understand where this person has been over a certain period of time. It is quite fun, but it can also be useful for the society.
This is another very recent story. Haven't been able to find the communication of this important pediatrist in Stanford. It is a nice case. Well, it is nice because every new case brings with it something nice to observe for the other scientists and researchers. So one infant got a cardiac arrest the same day she was born, and please give me a few minutes. This child had this cardiac arrest, and so the defibrillator was found, and so the defibrillator helped the child to survive. And the molecular analysis in the blood sample found the mutation in the SCN5A gene, which was not present as we saw later on in the family. No mutation was found in another test. So this questioned the test, but then it was repeated several times, and so the girl was, actually it was an infant who had a mosaic of 2 types of cells within the same organ. So how did they understand this? Well, they went to a single cell analysis expert in every white blood cell, and 33 of them showed a normality in the gene, and 3 were mutated cells. So similar results were also found in urine and skin cells. So the 3 layers of cells that in the embryo produce the 3 types of cells different. They originate from the same cells in the 2 we called embryo. So you can see that very early there can be modifications in the genome. Unfortunately, after a few months, the child once again had an irregular heartbeat caused by the defibrillator, but unfortunately, she had heart arrest, and the only thing they could do to save the child was to receive the heart transplantation. So So how was this case solved? They took the heart of the child had a new heart, fortunately, and so they found in several parts of the heart 5% of mutated cells in the right side of the heart and 12% of mutated cells in the left side of the heart. And so we can conclude by saying that the genome doesn't vary only from person to person. Unfortunately, I do not have with me the data of the variability, but we can also vary from cells to cells, and so we are probably all mosaics, in spite of what we believe is something very similar. I'm sorry if I will skip a few slides, but I have to be quick, and I have to show you the final part of my presentation. So the problem concerning gene mutations are very important because we can assess the function. They can be pathogenic, they can be likely pathogenic. Another is quite a fixed classification, but it is not a final one because unfortunately because we have several situations. So there are many cases of diagnosis of genetic diseases that we do also in a prenatal phase, and so we studied the functionality of mutation as we do in many laboratories, and in this functionality we have two things that we do. As the previous speaker said, we do a set of panels of genes related to disease, which are confounding. We are keen to do that on the basis of the diseases which are confounding. For example, in the cases of cardiovascular diseases, we performed 3 of them and afterwards 5. These are the types of results which were achieved, but I have to skip them because of the time. Here some mutations which were not known were found, so the mutation was found, but also a splicing was performed, and so the need to understand how the splicing produced an alteration of the messenger and so of the protein, and this is an electrophysiology study. We have a small electrophysiology lab which allowed us to say that for some mutations of the potassium-sodium channels we were able to understand whether they were pathogenic or not. This is the field of cancers, as the previous speaker said. There are several panels. We have almost 7 or 8 with several genes, and going to the conclusions. Otherwise, I will be. Well, the chairman will be very mad at me. So this is a study on cystic fibrosis, modulator genes. So we have a big experience. Professor Castaldo in our department has great experience in this disease. So you can we can say that we were the pioneers in the analysis of these modulators in cystic fibrosis together with other international groups. But now we would like to make a comprehensive analysis on all modulator genes. So this is the study, the functional study of some new mutations of BRCA1, BRCA2. I'm sorry, I'm going very quick, but well, all this allows us to understand precision medicine, which concerns the subpopulation that differs in their susceptibility to a particular disease. And so also biology or the prognosis of those diseases that they can develop can adjust to some specific treatments. I'm a bit against the use of precision and personalized in the same way. I prefer this expression, so I prefer to say individualized medicine, because medicine, it is a medicine that must address not only the patient to respond to the same drug because they have the same mutation, as it is happening in the case of cystic fibrosis, but also in the cases of other diseases and cancers. But it is very important because the individual has to be understood not only on the basis of the disease. I believe everyone agrees on it. Every individual has to be understood as a single individual who can have several diseases, and each one of us is different from the other individuals, just like the genome. And so these are the conclusions that I have already said during my presentation. I hope that those who study this matter, genomics, and its other aspects could be interested in what I've just said. Thank you.